You know, the world is absolutely teeming with animals. I mean, scientists have described over 1 million different species, and that number just keeps going up. So how on earth do biologists make sense of it all? Well, it turns out they have a kind of blueprint, a set of core characteristics that helps them organize every single creature, from the simplest little sponge all the way up to us. Today, we're going to explore that blueprint together.
So here's our plan. We're going to build that very blueprint, step by step. We'll start with the most basic building blocks of life, then move on to body plans and embryonic layers. After that, we'll peek inside at their internal design, and finally, we'll put it all together into a complete classification toolkit.
And really, this is the big question, isn't it? Because classifying all these animals isn't just about making some tidy list. It's about understanding the deep evolutionary relationships that connect all life. And the answer? Well, it lies in looking for these fundamental differences in how animals are built from the ground up. Okay, so our first stop is the most fundamental one of all, the level of organization. We're asking a really simple question here. How are an animal's cells actually put together? Are they just a loose collection of cells hanging out, or are they organized into something much more complex?
Think of it like you're building with Legos. At the simplest, the cellular level, you've just got a pile of loose bricks. That's kind of what you see in sponges. Then, at the tissue level, you start grouping similar bricks into walls. That's what we find in animals like jellyfish. Take it another step to the organ level, and you're assembling those walls into a room, like a stomach, for a specific job. And finally, you get to the organ system level, where you connect all the rooms to form a complete working house. Think of the digestive system or the circulatory system in animals like us.
All right, next up in our toolkit is the animal's overall body plan, or what we call symmetry. This is all about the basic shape of an animal and how all its different parts are arranged. And these two images show the two main types perfectly. On the left, the starfish has what's called radial symmetry, while the crab on the right shows bilateral symmetry. You can see the difference right away, can't you? One is kind of built around a central point, like the spokes on a wheel, and the other has a very clear left and right side. So here's the key difference. With radial symmetry, and the pizza analogy is perfect, you can slice it through the center from any angle and get two equal halves. Think sea anemones, jellyfish. But for bilateral symmetry, like a person or that crab, there's only one way you can slice it, right down the middle to get a perfect mirror image. Most animals you can think of, from insects to dogs to us, have this bilateral design.
Now we're going to go a little deeper and look at how an animal develops when it's just an embryo. The number of cell layers that form at this super early stage, we call them germ layers, is a huge dividing line in the animal kingdom. So animals basically get sorted into two big groups here. You have the diploblastic animals, which have two germ layers, an outer one called the ectoderm and an inner one, the endoderm. But then you have the triploblastic animals, and they add a crucial third layer, the mesoderm, right in the middle. And that middle layer, well, it changes everything. Let's look at a diploblastic animal first. You've got two main layers. The outer one, the ectoderm, becomes things like skin and nerves. The inner, endoderm, forms the gut. And in between them is this non-cellular jelly-like stuff called the mesoglia. There's no complex middle layer to speak of.
Now compare that to the triploblastic setup. See that red layer that pops up between the blue ectoderm and the yellow endoderm? That is the mesoderm. This third layer is an absolute game changer. It's what develops into muscles, bones, and most of our major organ systems. It's what allows for this huge leap in complexity.
Okay, so for all those triploblastic animals we just talked about, there's actually another level of classification. And this one is all based on whether they have an internal body cavity called a colum and what that cavity looks like.
So what in the world is a colum? Well, it's basically a fluid-filled space inside the body that holds all the organs. But, and this is the critical detail, a true colum has to be completely lined by that middle germ layer, the mesoderm. Let's take a look at what that actually means. First up, we have the columates. They've got a true colon, which you can see here is that white space. The key is that the mesoderm, that red layer, completely lines the cavity. It's on the outside near the body wall and on the inside surrounding the gut. That full lining is what makes it a true colon. Next up is the pseudocolomate. Pseudo, of course, means false, and you can see why. There's definitely a body cavity there, but the mesoderm doesn't form a solid lining. Instead, you see that red layer just in scattered pouches between the outer and inner layers. So it has a cavity, but it's not a true, fully lined colome. And finally, we have the acolomate. The prefix a means without, and that's exactly what you see. The whole space between the outer and inner layers is just completely filled in with tissue from the mesoderm.
There's no body cavity at all. It's a solid construction through and through.
So let's do a quick recap of that. Colomates, like annelids, earthworms, they have a true body cavity that's fully lined by mesoderm. Pseudocolomates, like roundworms, have a cavity, but it isn't fully lined, and acolomates, like flatworms, have no body cavity whatsoever.
So we've gone through the major criteria. Now it's time to put it all together and add the final features to our classification toolkit. Let's assemble the whole blueprint.
Two last really crucial features finish off our toolkit: segmentation and the notochord. Segmentation, you can see it so clearly in an earthworm, is when the body is divided into a series of repeating units. This is a brilliant evolutionary trick that allows for more specialized body parts. And then there's the notochord. This is a flexible, supportive rod found in animals called chordates. This structure is actually the precursor to the backbone in vertebrates like you and me, which makes it a really, really big deal.
And there you have it, the completed blueprint. By just asking a series of simple questions: what's its level of organization? What's its symmetry? How many germ layers does it have? What kind of body cavity? Is it segmented? Does it have a notochord? Biologists can systematically place any animal into its proper spot in the huge, amazing animal kingdom.
So the next time you see an insect or a jellyfish in an aquarium, or heck, even just look in the mirror, you now have the toolkit to understand its fundamental design. You've got the blueprint. The only question left is, what are you going to classify first?